Good afternoon, everybody. Welcome to, hi everybody. Welcome to the March webinar for HTML.net under the theme of genomics, which is going to be presented by Professor Kristen Happi, who is a director at SGED, as you can see on the first slide at the moment. But I'm going to be reading a bit of the abstract and a bit of his biography as well, which is a very long biography. I sent out an ad. If you want to obviously read more about him, you can Google him. It's very much Googleable, and you can find his publication, many publications on Google Scholar as well. So please do, obviously, after the webinar, give a bit of time to do that. Just a few guidelines or ground rules that we run with at HTML to make sure we don't disturb our speaker. I'm going to kindly ask that you type your questions in the chat box. So that's the first one. That you mute yourself. So if you know that you're not speaking, so some of you, I understand, are working from home and there are children running around, or some people are playing music in the background. So as to not disturb a speaker, kindly make sure that you mute yourself. Otherwise, I'm gonna have to mute everybody, so I don't wanna do that. So please do engage, type your questions in the chat box, as I said. And at the end, we're gonna have about 10 to 15 minutes where I'm gonna be fielding questions for our speaker, who's gonna be presenting on the title, Genomics, Characterization and Surveillance of Microbial Threats in West Africa. Before I go on, I just want to say Happy World TB Day. In 1882, I think, Robert Koch came out and announced that he had found the bacterium that obviously leads to TB. So most of you know about that as well. And I mean, we only have one vaccine at the moment. And I think everybody's saying that the clock is ticking. We do need to find a vaccine or a cure. And I think it's quite fitting that Prof. Happi is giving us this presentation today on genomics of characterization. So just a bit on the abstract, which I sent you as well. Identification and characterization of microbial threats, such as infectious bacteria and viruses from human to human or animal to human transmission, remains crucial in public health, most especially in globally connected. Diseases from the most remote villages in the world can spread to the most civilized city in under 48 hours because of ease in travel and doing business in the 21st century. Furthermore, West African countries have been adversely affected with several infectious diseases ranging from Lassa, Ebola, fever, Dengue, and novel human coronavirus, which has resulted in thousands of deaths. Here, Professor Happi and his group and co-authors applied an unbiased next-generation sequencing approach, also known as metagenomics, to detect and characterize a wide range of infectious diseases in the region from clinical samples such as blood, saliva, and nasopharyngeal people during different outbreaks. Can you please mute yourself so that we don't disturb this? Yeah?
Hi, thank you.
Hi, sorry, I'm gonna mute all. Sorry, because now I'm sorry. Yeah, I was distracted a bit.
West Africa.
I'm gonna. Sorry, I'm finding the.
No. Genomic characterization and
that was sorry about that. So that was one of the. I just muted all. So apologies about that disturbance. So just to make sure that we don't disturb a speaker like I just got disturbed now, I'm gonna make sure that I mute all of you all the time. And only at the end will I give you the rights to speak. Okay. Thank you. Sorry about that. Just a bit of the biography on Prof. Kristen Happi. He has an illustrious career. As I said, he's won so many awards. He's been around the world. He's one of the most important scientists, I'd say, in my mind, in Africa. And obviously his CV proves this as well. Professor Kristen Happi is a professor of molecular biology and genomics in the Department of Biological Sciences and the director of the African Center of Excellence for Genomics of Infectious Diseases, SHERLOCK for short, at Redeemers University in Nigeria. He holds a BSc in biochemistry from the University of Yaoundé, Cameroon in 1993, an MSc and PhD in molecular parasitology from the University of Ibadan in 1995 and 2000, respectively. He did his postdoctoral research in molecular biology and genomics at Harvard University School of Public Health in Boston, Massachusetts, USA from 2000 to 2003. He's also the director Directorate of the Research and Innovations and Partnerships, DRIPS, at Redeemers University. His research focus is on human genomics, molecular biology, and genomics of infectious diseases, especially malaria, viral hemorrhagic fevers, Lassa fever, Ebola virus disease, and others, HIV, and the novel human coronavirus, SARS-CoV-2. His current research activities consist of using innovative approaches that combine patient care, fieldwork, laboratory, molecular biology, and genomics methods for discoveries that have shifted the paradigm in clinical research and applications in parasites and viral diagnosis, parasites biology and genomics, pharmacogenomics and human genomics. As I said, he's done so much. So I'm gonna skip a lot of that because I'm looking at the time and I'd love for him to start. But just lastly, in 2019, he was awarded, deservedly so, the Human Genome Organization Hugo Prize as a recognition for his contribution in the field of genomics and infectious diseases in Africa. In addition, he was awarded the American Society of Tropical Medicine and Hygiene, Bailey K. Esford Medal in 2020 for his lifetime achievement in tropical medicine. I can continue reading more and more about his illustrious CV, but I'm seeing that I'm chipping away at the time. Without further ado, Prof. Christian Happi, thank you so much for taking time out to give this talk. Without further ado, the mic is yours, Prof. Go ahead and give your presentation.
Thank you, Pablo, and thank you to H3 Africa for organizing this symposium. I think it's a pleasure, and then it's an honor for me to present here today. And then what I'll do today is basically telling you a story and how we've been able to use genomics for the first time in Africa to address one of the major problems, public health challenges in Africa. So Christian Happi is my name. I direct the African Center of Excellence for Genomics of Infectious Diseases at Redeemers University. And this center is the WHO reference center for infectious disease research, genomics in Africa. And then it's also the Africa CDC, reference laboratory for the continent. And then what I will be talking about today is mostly, this is just giving this outline, how we kind of use genomics to cover many of the epidemics and then infectious disease outbreaks in West Africa. So, and the reason why we do this was basically because a few years back, we realized that genomic sequencing technology has evolutionized the way, healthcare and the medical research is being pushed across the globe. But unfortunately, many of those technological, both technical health and economic benefit, we're basically creating a huge gap between Africa and the rest of the world. And the reason for this is basically because we realized that, many Africans, did not have the knowledge or the training or the skills that were necessary to use genomics in order to address, the health challenges or to contribute to health development, globally. And what we did was basically to, first of all, think about how we want to use genomics as a way, to address, major problems, in Africa, while building the necessary local capacity and then transferring knowledge, across Africa. So in order to do that, we were able to, we set up a platform, and that platform is called the African Center of Excellence for Genomics and Infectious Disease. It is a multinational platform that is made of clinical and then academic centers across West Africa, but also collaborating with international partners like Harvard, the Broad Institute, universities, and then Illumina, and then many of these, the Variant Emerging Consortium. And then this list over the years has become very long. But then this platform is really built around, what I call long-term, research and training partnership. And what is our mission? So our missions are really two goals. First is, to create an academic and vibrant research environment, that transcends national boundaries, because we know that knowledge in Africa doesn't circulate. Brain circulation in Africa is very limited because of all the visa restrictions, but also create an environment where, Africans can conduct relevant, responsive, and then high quality translation and genomic research. You're going to see how we really apply this in the context of many disease outbreaks. And then these are the specific aims, that is building or developing research capacity for genomics in Africa by building a critical mass of well-trained African scientists that can, that can use genomic knowledge in order to address public health problems. And this specifically, that is control and elimination of infectious disease. And then in addition to that, we have other platform. But why did we, basically, in order to do this, we actually identify what is a research thematic that we should be addressing. And we looked at infectious disease, and then, in infectious disease, we realize that looking at fevers actually will be very helpful because most pathogens or most infectious diseases in Africa are actually first manifested by fevers. And then the idea is most of the time to identify what are the drivers of fever illness across the continent, because mostly in many parts of Africa, when people have fever, the first culprit is malaria. But we also know that many other pathogens, could be viral, could be parasitic, could be microbial, basically of bacterial. I mean, could be responsible for the fever. So the idea was actually to see how we can use this approach as a way to identify what are those drivers, and as doing this, we'll be able to identify the microbial landscape. And then through identification of microbial landscape, we could develop the right countermeasures. And then what I mean by diagnostics, vaccines, and then therapeutics. So this is the kind of stuff that approach that we've used now for this. But then in using this approach, we realize also that we're meeting, one of the critical needs for Africa. And this has to do with two things. One is education, and then the other one is research. The education component of this program that we've actually established is funded by the World Bank. So, in that we offer a lot of what I call a scholarship for masters and PhD in genomics and then as I apply to infectious disease, it's the very first program in Africa that is doing this. And then we also, conduct research related to developing countermeasures and then using genomics as a way to address, these public challenges on the continent. And then our ultimate goal is actually to develop what we call the next generation the next generation African pathogen hunters that will help, hunt the pathogens in Africa, but also to help develop the countermeasures in Africa and in collaboration with partners that are already willing. So, and this I will basically show you in the next few slides how really we transform outbreak response. Starting in 2014 in the Ebola outbreak. As you know very well, the Ebola outbreak started in some way in Guinea. And within that time, at that time, there was really no genomic facility on the continent or the capacity to actually conduct genomic research in the continent. What we did basically at the time was actually to bring together friends then that were thinking alike. You could see here is a meeting that I called in my lab at the time. And then with friends and colleagues, we actually sat down and they started thinking about what would be, what could we do in order to have the outbreak response in Africa? And we realized that diagnostics, was one of those major gaps. And because if you can diagnose a disease, then you cannot definitely manage it, you can treat it, and then you cannot actually create a vaccine for it. So what we did was, in Sierra Leone was the fact that we were able to use, for the first time, genomics as a way to understand actually how the virus was spreading across West Africa. In the first 3 weeks of the outbreak in Sierra Leone, we identified 35 suspected cases, confirmed 14, and then went on in the first 3 weeks of the outbreak, generated the first 99 full-length genomes of the Ebola virus at the time. What really made our own work special and unique was the fact that for the first time during an outbreak, we were able to generate genomic sequence data in Africa, but we also made this genomic sequence data openly available to the international community, to the scientific community, so that those that were interested in using this information for vaccine development, drug development, and diagnostic development, could use it. So in real fact, we actually pioneered what we call open data access. And we're very happy that that process that we started in 2014, is today the real standard. Today, you can see the GISET as a platform that the whole global scientific community is using in order to share data. We pioneered this in 2014, and we're really glad that this kind of this type of this really was pioneered within Africa, and then we led this effort. What we did at the time was really incredible. That is basically from sample collection to submitting the data to NCBI, 10 days. And that really, we made this data available. And then our publication could come back until 4 or 5 months after. So which means we're really not out to get fame, we're really out to share information instead. We use this actually to, first of all, this is a first demonstration. So you can see here within the case of Sierra Leone here, where we demonstrated, use genomic data during the outbreak to say that the outbreak or the epidemic in Sierra Leone or West Africa was due to sustained human-to-human transmission, and that there was no additional introduction from animal reservoirs, and then no new imported cases, based on genome and viral diversity. And you could see here where within the case of Sierra Leone, and if you look at here, where we have different incursion of different lineages of the virus, 4 different lineages over time. But then by June 16, you could see the lineage SL3 actually taking over from that country. And it really proved afterward that this lineage was more efficient in transmission, highly transmissibility, and then was also a little bit more pathogenic. So we use this information not only to identify the different SNPs, to identify overall at the time that we had about 325 single nucleotide polymorphism across, but then 27 of them really were non-synonymous, and then they become a little bit more fixated in the genome. But then more interesting, for the first time, we were able to use genomic data actually to show transmission spread, and also how the virus can move, spread over time, and then transmission from country to country, from county to county. And then this really was the very first time we had this demonstration. So if you look at it, you could see that the board here, you could see how using genome data, we're able to show how the virus was spreading from place to place. And this is across the whole of West Africa, and then across Sierra Leone, and then from Sierra Leone to Liberia, and then Liberia to Guinea. So really demonstrating genomic epidemiology in action, and then having this data provided in real time. So we actually had this study published in Nature a few months after that. But what was most important was the fact that we were able to use genomic data and translate genomic data into tools, and which is in the case with point of care diagnostics. So we realized that the turnaround time for diagnosis during the outbreak was such a long time, and that was also one of the drivers of transmission of the epidemic in most West African countries, because when people left their home with a fever that was in malaria, went to the Ebola testing center, they got infected there because it took such a long time in order to get it resolved. So what we did was basically translating our genomic data into point of care diagnostic, and within 4 months of that outbreak, we were able to establish, the first Ebola rapid diagnostic test that was approved by the WHO and then the USFDA under emergency use authorization. The introduction of this test really was actually a major turning point in order to contain this outbreak, in West Africa. So the case of Nigeria, for example, a country of more than 200 million people, were able to use speed and accuracy, to contain this outbreak. In 93 days, we diagnosed the first case within hours, and then we used all of this data in real time, and we were providing the information to the Nigerian public health authorities, and they were working in a very laser-focused and laser-pointed manner, so that in a very surgical manner in the way they're interacting. And that's really why Africa, Nigeria really went back into the history in the annals of medicine and public health, because this was a country that has a potential to actually bring down the African population to its third. But fortunately, because of the approach that we use, and then because of the fact that they accepted this technology at the point, they were able to contain this outbreak in 93 days. Nigeria ended with 20 cases, and then 12 deaths, and then 8 survived. And this was really unprecedented in history. So, but then the point is, talking about Ebola, there's another disease that is plaguing West Africa, and this disease is called Lassa fever. This is a virus that was discovered in Nigeria almost 52 years ago, but then by about 10 years ago, this virus was rarely, diagnosed in Nigeria. Not only we set up the first molecular diagnostics in Nigeria about 12 years ago, but we also went further to actually do the first genomic sequence of this virus in the world. So by sequencing this virus, we basically just made available this information, this great knowledge body of information that enabled and accelerated, a vaccine for this disease. 10 years ago, this disease was rarely diagnosed. I can tell you that 10 years ago, we set up the first diagnosis. 5 years ago, or 6 years ago, we set up the first genomic sequences. And I can tell you that within the 6 years and now, there are 3 vaccines that are actually being tested in West Africa for the disease. Thankfully, thanks to the fact that we were able to actually generate the largest genomics data set, to this virus. So showing here the firepower of genomics when it comes to, to develop countermeasures. So in doing that, basically what we found was the fact that even though this disease I told you was described the first time 50 years ago in Nigeria, we found that this disease actually was a disease that has emerged from present-day Nigeria about 1,006 years ago, was circulating within Nigeria for 600 years, moved out of Nigeria about 400 years ago westward. And then taking advantage again of the genomic sequences, we're able to actually translate, that genomics into countermeasures, and then basically developing what we call a pan-Lassa rapid diagnostic test. And this test actually enables you to get a diagnostic in 10 minutes. And this has been very helpful in the region in order to understand clearly in order to detect actual detection of cases. And this work was done in collaboration with my colleague for disability, Christian Anderson. So we had this publication in the South, and that has been very rewarding to see this. But most importantly, is that when we do this work in the field, then you realize, as I said before, that you have, fevers, and some people actually come to the hospital. They present with cases that are similar to a barrage of pathogens. But when you go on and then diagnose them by the conventional method, you realize that all those diagnoses are negative. Then the question that is often asked is, what is responsible for the fever? Is it, for instance, a virus that is mutated, or is it, due to a new pathogen that we don't know? So in trying to address this question, we introduced in 2015, the very first time in Africa, using microbial metagenomics in order to identify, pathogen in clinical samples. In doing this, for instance, we're trying to address one question. What is, for instance, is the microbial landscape? I mean, what is the microbial landscape? I mean, within a given region, and also is it that we can actually identify new viruses or new pathogens that could be responsible of disease that are similar to the ones that we know? And interestingly, in the process, we basically just try to identify, do the case control study, try to identify what are the drivers, the pathogen that are driving febrile illness. So here you could see the case where we have people with febrile illness and those that are healthy in court. And then what was interesting in the process is that we used microbial metagenomics for the first time to identify two brand new viruses that are called arabnoviruses. And these arabnoviruses were found in people, here you will see in healthy control, but then by the time we went back to see this patient, we realized that they are taking antipyretics, and that's the reason why at the time where they were found negative. The two new viruses that we discovered at the time, we call them epomavirus 1 and epomavirus 2. You see them here. But then these viruses we found share similarity, for instance, with another virus that was involved in discovery. Involved in a viral hemorrhagic fever in the DRC, in the Bakongo, called the Bakongo virus. So these two new viruses, as we see today, are very important. Now, moving fast forward in 2018, in Nigeria actually was facing a huge public health emergency, because at the time, within 4 weeks or 8 weeks of the in the early phase of the year, in the early part of the year, the country found itself with more than 2,000-fold increase in terms of more than 200-fold increase in terms of Lassa fever cases. That really was creating a lot of problems. Then there were a lot of unanswered questions. The question was, are they dealing with new variant, or is it that the surge that was observed in Nigeria was due to human-to-human transmission? So what we did at the time was actually to take a sample and did real-time metagenomics in order to address the question. So we collected data, the sample, and then performed metagenomics data right on site, and eventually we were able to provide the answer. So, and this was really done by this great team in my lab there, but this is an old picture, so I think this achieved a great deal. We came up with very clear answers that there was no evidence of particular new viral strain. There was no evidence of increase or sustained human-to-human transmission, but that the surge that we saw in Nigeria at the time was due to multiple introduction of genetically independent viruses, of non-lineages, that is lineages 1, 2, and 3, from animal reservoirs. So we had this study published in the New England Journal of Medicine at the time. But what was interesting about it is the fact that we realized at the time that Nigeria was dealing with more like 3 different epidemics. One different lineage circulating in the north, one lineage circulating in the east, and one lineage circulating in the west. And that really was very revealing. And the more interestingly, we realized that all of these actually were transmitted from different organs. So, and that really, we got credit, from the WHO and the Nigeria CDC for this, because we're able to answer very important questions. But then by doing this, for instance, we were able to understand that it was mainly about rodent, and we set up a very, very important, large, study to study the Lassa fever diversity in rodents in Nigeria. But what it was interesting was for the first time in the history, of infectious disease, we were able to establish, for instance, the direct link using genomic sequences from rodents and in human, to establish, a direct infection between viruses found in the rodent and then viruses in the human. And that really was very helpful. And that has been the very first demonstration of using this technology as established socially. But then the question is, knowing this, we really think about how you want to use this technology, not only the sequence data. What we did was basically to use sequence data to create a better technology or better diagnosis, because we all know what the challenges are with antigen captures. But we use what we call gene editing technology, use a CRISPR, technology as a way to develop a more sensitive, assay for Lassa fever. And that really is also something that we translated for the case of COVID. So we basically just use the sequence data, identify sequences that are specific to different lineage, consensus to Lassa fever. So then design guides RNA to identify those, to target those sequences. And effectively, with the guide RNA, we should be able to bring together like molecular scissors, you can nucleate the restriction in the nucleases, and then tag that along, with reporters. And then with those reporters, you'll be able to have, you can have a signal when it is Lassa fever. It's just exactly as you see there on the on the picture. So, and then this is really a big report in Nature, from our lab when Amy Maxon was embedded in our lab and really wanted to see this technology. And here you could see a student, Victoria, in my lab, really conducting this experiment. And you could see the team in my lab here running these things on site. That was the first time we were using gene editing essentially to address something like Lassa fever. Another example is basically how, for instance, in 2018, toward the end of 2018, there was an outbreak of a mysterious disease killing school children in the southern part of Nigeria. All tests, when you talk about all infectious disease tests, proved negative. So the Nigeria Center for Disease Control sent the samples to our lab. We used microbial metagenomics. This study was led by Victoria in my lab, and then with that, she was able to uncover an undiagnosed outbreak of yellow fever. And then this yellow fever, as you see here, for instance, this is a 2018, and then these are the other yellow fever lineages circulating in Africa. So it was really an urban transmission, but then what was important about this is that this lineage or this strain of yellow fever that was discovered by our team was actually very different from the other lineages that were circulating in Nigeria for the past 92 years. And this information was given to the Nigeria Center for Disease Control within 48 hours of collecting the sample. We generated this data, they declared an outbreak, and they were able to contain successfully that outbreak, in the southern part of Nigeria, saving thousands of lives if this outbreak could occur. This is another example where we basically just use again microbial metagenomics as a way to identify a new, enterovirus B. This is a case of a child in Lagos who was presenting signs of viral hemorrhagic fever. He was taken to hospital. The child, 2 years old, is dumped in the hospital. The parents disappear. The whole hospital is in crisis. They don't know what to do. Then the sample are sent to us very quickly. We're able to identify that. We're able to say that no, it is not Ebola. It is not that is not the virus that we know. We identify this enterovirus B into these children, in this child, and the information is shared with public health authorities. Fast forward to COVID. So when COVID started, the first case of COVID in what I call in sub-Saharan Africa actually was in Nigeria. So, and then once this happened, the Nigerian Center for Disease Control sent sample to our lab, and we did something that was unprecedented. Within 48 hours, we were able to generate the first genomic sequence of SARS-CoV-2 in Africa, and then share this information with public health authorities. But then this information was very important. So you could see here, for instance, this was the very first SARS-CoV-2 on the African continent, and this was done at the speed of light. This really was very important because that really provided information in terms of importation of the case, and also showing clearly that it wasn't a case it wasn't a local case. But this subsequently, what we did was basically to use, genomic epidemiology to map, the strain and lineages circulating in Nigeria. So far, we've had over 501 genomic descent, and then this gives you an idea of where these are coming from. Nigeria is a country about 37 states, and we've done this about 27 states where we have the samples. So this gives you an idea of the distribution of these cases over time and space in Nigeria, but also give you an idea of how you could see over time the introduction and then when new lineages occur and then when they spread. So today we have over 55 different lineages circulating in Nigeria, but among the 55 lineages, we have just about 2 variants of concern. So using genomic epidemiology and then extreme, and then this is really done by great guys in the lab, by Paul and Edo, really using this as a way to show this in real time. If you see how this is, you can see the live transmission of this in Nigeria, and this is how they've been able to use this over time to demonstrate to the public health authorities what are the hotspots, and then where the spots where the disease is actually, how the disease is spreading. As you can see in Nigeria, for instance, you can see Lagos and everything here. These are the really hot spots, and then this is where the government have been concentrating over time. But this really is very important because this is, you can see here, this is where genomic epidemiology in action. So I mentioned this before. So, but then what was also something very important to mention here is the fact that during the lockdown, we were able to show clearly that even though the government lockdown, you could see, viruses. This is the beginning of the lockdown. You could see in many states in Nigeria, just few, but you could see how, viruses were moving from one state to the other. So this really showing clear that the lockdown wasn't working, but then information given to government authorities just using genomic epidemiology to track the viruses, then they were able to develop, a different strategy. What we found in this case is that during the day, the lockdown was very strict, but then in the night, people were flaunting the lockdown and they're moving viruses from place to place. And that really is, again, showing how the genomic epidemiology can help you to track, the spread of infection over time. Then this is another example whereby the DC14G mutation, for instance, that we're one of the first group in the world to report this mutation. And we've been using this to track this mutation over time. And I can say now that over maybe 90% of the infections circulating in Nigeria have this particular variant. This was the very first variant of concern of SARS-CoV-2 that was actually described, in the world. So this variant is not just peculiar to Nigeria, it's actually spread across Africa, and then you can see this over time. But that's really shows how you can use genomic epidemiology to track. So we're not doing this just for the purpose of just looking at the spread, we're also looking at what is the potential effect of all these mutations that we're seeing across the genome of vaccines. So these are the latest data. When you look at the China CDC and primers, you look at the US CDC, the Pasteur, and then the Charity and primers. So this really show you that if you really run in tests with the China and CDC primers, then the probability that you're having a lot of false negative is very high because the mutations are accumulated where the target primers are, is very high. So that really also is very helpful in the sense that we monitor the tools and the diagnostic tool kit that we're using. But then I will probably just push it here again by talking about, the second wave, of COVID in Nigeria, because around Nigeria, just like most places in Africa, the cases dropped dramatically between probably August and then October, but around November, December, we started seeing this huge spike. So the question was, what really was the responsible for the spike that was happening in Nigeria? So we really went on quickly and then tried to see exactly what was going on. I think there's a mistake. What was driving this thing? What really happened in this case was the fact that we realized in Nigeria that we just discovered, or in our lab, that there were two different lineages. One lineage, a new variant that we call, but we don't call it the Nigerian variant, but a variant that emerged from Nigeria and spread across the world, which is the B1.525 variant, and then also the B117 variant that we actually identified across, many states in Nigeria. And that basically was probably what we're driving, and then the B1.525 is now a variant of concern, but then that's really also explaining how we could use genomic epidemiology. So basically just giving you an idea of how we need to use it for the context of COVID to identify B117 lineages in Nigeria, identify the emergence of a new lineage called the B1.525, which is now another variant of concern, but then also establishing the fact that we have multiple introduction in the country from different part of the world, evidence of community transmission, identify all this variant, and then the work is ongoing. But then the question that you ask often is, how do you use all this information? What is the benefit of just doing genomic sequencing? Is it just to guide public health response? The additional benefit of it is the fact that we're able to use this information in collaboration with colleagues to come up with a very rapid diagnostic test, which is the very first generation, what we call nucleic acid-based test that is actually on lateral flow. That is like on paper strip. You can identify, nucleic acid. And this within 5 minutes, you can actually get your, depending on the CT value, but it goes from 5 minutes, you can actually obtain, your data. But when your CT value is very low, you have to incubate for a longer time. So within 5 to 10 minutes, you should be able to get, 5 to maximum 90 minutes, you get result. What happens for most cases that with a low CT value, you can get this result within 5 minutes. So, and what is interesting about it is that we have two versions. One version that is on a paper strip, we also have a plate version, so for that is increase your high throughput. So there were a whole TV5 documentary and NBC documentary about this test that we actually developed with collaborators. So this basically just giving you an idea and see live how the readout of this test in our lab. What is good about this is that this test will really, can be applied to many other diseases. And then basically from saliva, you can obtain many other, you can detect many other viruses. What is cool about this is that not only you could detect, develop, a new test very quickly in case you have a new virus, but what is important is that within a week, you can go from discovering a new virus and then developing a new test. So we also have a multiplex, we call it CAMIN, which is a combinatorial array of multiple tests here. So we can do this test in massively multiplex, and then therefore detect over 400 or 500 pathogen or even 1,000 different pathogen in one single chip. Then you can also do multiple times that, the multiple thousand in a single of the same pathogen in one chip. So that basically the goal for us is actually to do, to develop what we call a chip that can enable us to identify, known and unknown pathogen, causing, fevers in West Africa. And then this is a gene-based diagnostic panels, that we can actually deploy based on extensive sequencing. And that these are some of the panels that we have currently in our lab, but then this is really where, the future is going to hold. Now, I will end up this talk by just bringing you a new concept. So this is a new, and for some of you probably have heard that, before, early in 2020, we were just given this great grant that is basically to start, developing, setting up a platform called SENTINEL. And SENTINEL is a pandemic preemption and response platform that we were setting up. We started this without knowing that we're going to be having COVID. And then COVID had really accelerated this platform. This platform is built on two major pillars. One is detecting known and unknown pathogen in real time. Second one is connecting, this result in real time to public health authorities. And then the third pillar is actually to empower the frontline health workers to use these technologies and integrate them in their everyday activities. So, and this really is built on the system that is on the spoke and hub approach, hub and spoke approach that we basically established in Nigeria. We have a hub and then we have the spokes, and then among the spokes, we have all these villages that we're actually supporting in order to do this. The idea is we're piloting this in Nigeria, then we're going to be replicating this in West Africa and then also in the DRC, in the next few years. So, and overall, the goal here would be actually to build what we call a community of scientists, researchers, and clinicians that actually could help not only to translate genomic knowledge, into not only high throughput diagnostics and serology method, but also to point of care diagnostics. And then by doing this, we will create basically what we call, a feedback loop, whereby working together, we can create all of these diagnostics and then develop, connecting this to the public health system, and then eventually create what we call an early warning system. This early warning system that is Sentinel actually is built on these three major pillars that I mentioned before, but then the goal here is actually to create a safer world. Imagine for a second, if you had the ability to diagnose, SARS-CoV-2 early, then we would have actually prevented the world to be in the pandemic. So we're building a system that will enable us to prevent the next pandemic, that will enable us to prevent the next disease outbreak. So at this point, I really want to thank you for your attention, and I really thank all these incredible group of young Nigerians and young Africans, that are working behind the scene, generating all this data, and then making this presentation possible. So thank you very much for listening. I also want to end it by thanking all our founders, and then all our partners, and all our collaborators. Thank you.
Okay. Thank you so much, Prof. Happi. You're doing important work and amazing work, and thank you so much for putting Africa on the map, scientifically so. And I'm grateful for this talk. This was fantastic, really incredible. Someone from the comments saying, "Thank you so much, sir, fantastic." So what I'll do now, we have a bit of time. I'm gonna now allow people to be able to speak. However, please raise your hands. Okay, I see there's a hand. Oh no, someone is clapping, I think. Let me open to see who's doing what. So please raise your hands if you have a question, or like tap in the chat box, and then I will relate that to Prof. Happi. Segun Fatumo saying in the comments, "Thank you very much, Prof. Happi, for this great presentation. This is a question, are there SARS-CoV-2 genomic variation that you found to be associated with low mortality rate of COVID-19 in West Africa?" Prof. Happi?
No, we haven't found that. I think if you listen to my presentation, you found only 2 variants, and those variants seem to be associated with high mortality rate and then high disease severity instead of low mortality rate.
Okay, thank you for that. Okay, people are really grateful for this outstanding presentation. Everybody's agreeing with me that this is timeless and very relevant and incredible as well. As I'm saying, you do have a right to ask questions. Please do raise your hand for me to be able to see who you are and give you the right to speak. again, you can then obviously type in the chat box as well. Professor Faisal saying many thanks for the excellent presentation. Bernard, you've raised your hand. Go ahead and ask your question.
Hi, thank you so much, Professor Christian Happi, for the fantastic talk. I was just curious to find out, have you identified more than one lineage from a single individual using these assays? And then the other one is just a comment to say that you show that there's an increase across the continent from October for the second wave. Don't you think this is driven by opening up or sort of reducing the restriction in movement? So opening up recreational parks and places of enjoyment, because it coincides across the continent. And if you look at data from, say, something like Google Maps, you can actually see a rise in recreational activity that correlates with what you're saying. Thanks.
Yeah, I do agree with you that opening up and then activity will create more exposure, but it's also, exposure does not necessarily mean that you're infected by disease. And then also, the fact that you're infected with this variant is actually clear that this variant infects more cells and then more and faster than the normal or the wild type or the original variant. And studies in vitro and have demonstrated clearly that when you get cells infected, then this variant is said to infect more cells than the original variant. So yes, you have exposure, but then also the fact that you have the number of increasing number of cases actually due to the fact that this variant seems to be more efficient in terms of, they have increased transmissibility power than the original variant. So I think it's a combination of both, but it's also the fact that you have a variant that is more efficient. It's not just for COVID. We actually showed that also during the Ebola outbreak in West Africa in 2014, 2016, that once we had that variant that actually had those mutations, they become more efficient in transmitting. So, and that really is also one major force behind the major driver behind the numbers that you see, or behind the spike or the surge that you see across Africa. In most, especially in the case of Nigeria, for instance, whereby it's not a situation whereby the restriction were people were not respecting the restrictions, but then we just saw this sudden surge. And it's more like you have a normal situation or things are just the same, but the number of cases actually just spike up, and then you wonder what has changed. And when you go in and then look at it, and you realize that a whole lot, it can be driven by these variants of concerns. Over.
Sorry, and what about the identification of multiple lineages in one individual? I'm asking this because if you go back and look at outbreak data, for example, Ebola, where primarily the researchers at the time were just looking to see whether the pathogen is present. But then if you go now and use more sensitive detection algorithms that look at metagenomic data, you actually see that they only reported one, say, strain variant or lineage, and missed out on the fact that some individuals could have had a mixture or a cocktail of lineages. So have you identified this in the current data set?
No, we haven't really looked at that kind of a very kind of granular type of data, but then I wouldn't preclude that. I think it's very possible. And then multiple infection is absolutely, or infection with different clones or lineages is very often, found in, especially in the case of viral disease and even parasitic disease. So I think even bacterial disease, I think it cannot be far from that. I think that's really a very strong possibility, especially in the case where you have, sustained transmission, and then people move from places to places. I think it's very, very highly possible.
Thanks.
Okay, thank you so much for that engagement and questions, Bernard. I see there's a comment/question from Prof. Faisal. We hope we could see similar work in other regions of Africa, which allow making comparison across Africa. Did you try to compare the results of West Africa with other regions within Africa? That's a question from Prof. Faisal. Prof. Happi, do you have an answer for that?
Oh yeah, I mean, we have, I mean, with the Africa CDC and the Pathogen Genomic Initiative, you know, we have two major labs in Africa, the reference laboratory, CRIPS in South Africa, and then my lab. We're not only doing supporting the country, but we're also supporting other African countries. CRIPS is doing a whole lot in Southern Africa, so we're doing a whole lot in Western Central Africa, and then all the way to the Horn of Africa. So we basically currently we're sequencing samples from Somalia and then from from Sudan. So we basically have seen, you know, that, and then we've seen, like, we also did some sequencing in Central Africa, Cameroon, and then one other country. I mean, to be very specific, and we see, you know, different patterns. But you also see a lot of what I call intra-African, you know, transmission going on. I think it is clear that, you know, these are not because of migration. So we're seeing a lot of movement of viruses from country to countries, and then that is very clear. I mean, anytime I'm with my friend Tulio, we see also, we look at data from South Africa, we see from Southern Africa, you see a whole lot of movement of viruses from place to place. I think it's only normal. But then this data become very important, you know, in order to set up the right set of policies, and also in order to track, you know, infection and then the spread of these variants of concern, especially.
Okay, thank you for that. Faisal is agreeing with you there as well. So there's a few more questions. So I'm looking at time. We have about 10 minutes left. So if you have a question that's pressing, please do ask it now. But I'll read the questions that are coming up in the chat box. Ademola Adenini says, what is the future of genomic sequencing in curbing tropical diseases? That's from Ademola. Prof?
Well, I think the future actually for genomic surveillance is the future for public health, you know, and disease surveillance is going to be genomic surveillance, because with that you have a deep dive into what is circulating. You have a deep dive in monitoring, see how the viruses are changing, and if they become viruses of concern, then you become, you know, then you know how to act. And I mean, I think this technology has to be spread. This technology has to be democratized, and it's very important. And not only for human disease, but it's also for animal disease. And I think, you know, what the One Health approach here will be very important. And then that's as part of the Sentinel platform that we have developed, the early warning system that we developed. We basically at this point integrating, you know, animal disease surveillance into it, because I mean, we've seen clearly that most of the pathogens that actually have pandemic potential actually often spread from animal to human. So I think it's very normal to start thinking about an integrated and a holistic approach toward disease surveillance. So, and that's really the future of genomic surveillance, or the future infectious disease control, maybe highly dependent on genomic surveillance.
Okay, thank you so much for that, Prof. There's another question from Frederick Mubegi, who's saying, is the second wave driven by residual variants from the initial infection of new variants that have either evolved within it or coming in from elsewhere? Have you tried community level epidemiology using, for example, sewage? That's what Frederick is saying. Prof?
Yes, we definitely have clear demonstration that, you know, the second wave was due to circulation of variant from I mean, first of all, potential importation of variant, emergence of new variant as I demonstrated here, then establishment of those variants to community transmission, and then that result exactly in the way that you see. So it is, and then those variants, as we say, you know, are normal biological process. They definitely, because the virus is bounding should be mutating, and then the virus is under different selective pressure. So you expect to see, you know, this variant emerging, but then once you have the variant emerging, so the issue is often, if you are dealing with the variant of concern, then that becomes a problem. There are, I mean, as I mentioned before, presently in Nigeria, there are 55 different lineages, or you call variants circulating, but only about 3 of them are what we call variants of concern.
Oh, okay, that's interesting to note as well, Prof. There's another question. Sarah, I would like to know if ACEGED has internship opportunities for undergraduates interested in modern genomics. So this is putting you on the spot. Are there opportunities for internships for undergrads that want to work with you guys?
Oh, yes, we do have opportunities for undergraduate internship. The thing is that it is highly selective and is always filled up. We have a long queue at the moment. I can tell you at the moment that we have so many interns in the lab, and then that is very, very challenging with us. We hope we could accommodate everybody, but it's just that, you know, the pressure is just so much. We have these young people that apply a year or two ahead, and really we somehow we will overwhelm at the moment, but we're hoping that when we move to our new facility, which is a larger, much more larger facility, we'll have the opportunity to get more interns.
Okay. No, that's fair, Prof. Adegite, just look at the last slides. There's an email address here, use it. There's a website, keep on checking for opportunities if they open up in future, because as he says, they might be moving soon. Olabode has a question, Prof. Really awesome presentations there. Just a quick question about the SHERLOCK technology you mentioned. Really interesting stuff. I was wondering how field deployable it is vis-a-vis the need for sample preparation, perhaps incubation?
With the new SHINE technology, actually, my graduate student, Jessica, she's probably better to talk about it than myself, but I can tell you that you don't need incubation, you don't need anything, just saliva, and then everything is at room temperature, and then you get your result, like bang. So it is clear that it is very innovative in terms of technology, and then it's very promising. Imagine multiplexing these technologies and then multiplexing these assays in one single test. That's exactly what we've done, for instance, with the Carmen test. So if you multiplex this, it means that for every sample that you get, you can actually screen for hundreds of viruses in one single test. And that's basically what you do. You don't need like a high validity technology. You don't need a lot of investment. You don't need to train somebody. It's just like a pregnancy test. But the only difference with this is the fact that the pregnancy test, you're looking for antigen. In this case, you are really looking for viruses, nucleic acid, and then you have a reporter, so you're seeing it. So I really, really think that this is what the future holds. And then this is a point of care diagnostics. So really you do not need for one version. That is a version to the lateral flow, is a point of care diagnostic. If you wanted to do it in a larger hospital setting where you have many patients, we also have a multiplex version of it where you could do this in a 96-word plate. So I think clearly, and then all of the things that I'm talking about here are published already. You had all this published in Nature a few months back both for Lassa, for Ebola, and then for SARS-CoV-2. So we basically have all of this demonstrated and then published out there. And they are sensitive as a qPCR. So what is really cool about it is the fact that you're matching PCR sensitivity, and then the cost, imagine, is actually like 100 of what a PCR will be over.
Okay, thank you for that, Prof. And another question now from Ademola. Does the different variants consequently mean different drugs for each variant?
Not necessarily. I think it all depends on what your drug is, where your drug is targeting. If your drug is targeting where the variant is mutated in a way that it can affect your drug, then or your vaccine, then you have a problem. So, but as long as your variant has not mutated in a way that it affects your countermeasure, then you are good. So you can imagine, we were talking recently about the B1351, V2 in South Africa, for instance, having mutations. I mean, like the same type of mutation that we saw in the B1525 in Nigeria, which is basically that the E484K mutation that you can see. And this is really where it's being targeted by neutralizing antibody. Once you have that, for instance, those mutations, then it means that the area where the neutralizing antibody could target actually is further forward, and then that becomes a challenge. And in that case, you can have an immune escape, variant. So I think, that's what becomes a concern. But as long as, the mutation or the changes that you see in the variants do not affect, your countermeasure, the target could be either diagnostics or, therapeutics or vaccine, then you're good to go. And that's the reason why we need to be monitoring, as much as possible.
Okay, thank you so much for that, Prof. Looking at time, we have about 2 minutes left. So if there's a pressing question, quickly raise your hand. Or if not, obviously, as you can see on the last slide now, you can email Prof. He responds. Although he's very, very busy, as you can tell from the research that he's busy doing now, and with everything going on, so many pandemics at once. If you have a question now, please do raise your hand or tap it in the chat box, and I'll read it to Prof. But otherwise, you can obviously follow him on Twitter. Kristen_Happi, or follow HDR on Twitter as well, where they're constantly putting up their publications and information about their events that they're running. And on my side, this is also running live on Facebook, and I'm busy recording it as well. So if you have if you missed some parts of the presentation, do not worry. I'm going to be putting it up on YouTube and sending out the link to everybody who attended. And we put it up as in on YouTube, as I say, so you can easily search it for yourself as well, even if you don't get that email. But we also then put it on social media as well. So thank you so much for attending. I see someone, Frederick, asking about sewage genomics. I don't know if you want to clarify that, Frederick.
Okay, so I feel like there's some people who just go like collect sewage material or any other effluent from the sewage and use that as a means to perform the detection of whether you have viruses, because you have shedding up to like 3 days or so. So you could detect like the presence of a virus in some community, or like based on the line that where you collected your sewage material, and use that for genomic detection or epidemiology, and just know way in advance that you will be having like COVID in a certain region. So have they tried that in their studies, or they just dependent on just getting samples directly from the people? So that's...
No, we haven't tried sewage genomics. So I think we've basically been doing a whole lot of genomic from human samples. And I think I've read a whole lot about that. And then I think it's really also great and promising, because it really is important to think about the different, areas where you could actually have potentials, transmission. So, and that's really is very important as well.
All right, thank you.
Okay, so it's just 4 p.m. now, and I'm gonna kindly ask that we close today's webinar. Thank you so much to all of you for attending. Prof. Happi, you were brilliant. This was informative. From SHERLOCK Technology, we're really grateful for your time and expertise as well. We'll be in touch. And so for all of you, I'll be sending another announcement about another webinar that's going to be taking place in April, the last week of April. So just be on the lookout for that on Twitter, on Facebook, or on our mailing list as well. So if you're not part of that, let me know. Prof. Happi, thank you so much, and enjoy your day, everybody. Thank you so much.